Hello, my name is Sungmin Han.
And my name is Michael Miller. We're at the University of Alabama at Birmingham in a baseball field to tell you about our recent paper in this issue of Developmental Cell.
This paper is part of our ongoing effort to understand the role of an unusual cell signaling mechanism in amyotrophic lateral sclerosis or Lou Gehrig's disease.
So Lou Gehrig was a famous baseball player for the New York Yankees who developed this disease during his career. So ALS is characterized by atrophy of striated muscle and degeneration of motor neurons that control movements such as these. Nice catch. Now let's go back to my office where we can talk a little more. So this project is part of a collaboration with Hugo Bellin's lab at Baylor College of Medicine and Hiroshi Suda, who is now at McGill. So it started in 2006, but some of the basic ideas go back to 2001 when I was a postdoc in David Greenstein's lab at Vanderbilt. So we showed that C. elegans sperm secrete a 126 amino acid protein called the major sperm protein, or MSP, to promote oocyte maturation, which is shown here and involves muscle contraction, cytoskeletal rearrangements, a cell cycle transition, and metabolic changes. MSP folds into an IG-like structure that is found in a human protein called VAP-B, or ALS8. Now VAP-B also has a coiled coil motif and transmembrane domain. Neither protein has a signal peptide for secretion, and VAP-B is anchored primarily in endoplasmic reticulum membranes. In 2004, the Zatz lab showed that a proline to serine substitution in the middle of the MSP domain was strongly associated with ALS in a large Brazilian family. However, the role of VAP-B and the pathogenic mechanisms of ALS were not well understood.
Together with the Bellin lab, we set out to test the hypothesis that the VAP-B MSP domain was a secreted factor important for the motor neuron function or survival.
In 2008, we showed that the VAP-B MSP domain was cleaved from the transmembrane domain and secreted by certain fly, worm, and human cell types. Importantly, the P56S mutation acted as a dominant negative and inhibited secretion. So this suggested that secretion could be important for ALS pathogenesis.
In this developmental cell paper, we identified a conserved role for secreted VAP-B MSP domains in regulating striated muscle energy metabolism.
In time-lapse videos of wild-type muscle, the mitochondria are stably docked and undergo fission and fusion, but mostly within individual arrays. In contrast, mitochondria in VAP-B mutants form thin, highly branched, and mobile networks that are mislocalized. So we show that motor neurons cleave the VAP-B MSP domain from the transmembrane domain and secrete it into the extracellular environment. Here it acts through roundabout and LAR-like receptor tyrosine phosphatase receptors expressed in muscle. MSP downregulates LAR signaling, keeping ARP23 dependent actin nucleation activity at muscle I-bands. This directs mitochondria to the I-bands, promotes elongation along I-bands, and increases energy production. In VAP-B mutants, secretion is inhibited, LAR-like receptor signaling dominates, actin remodeling occurs, and mitochondria move away from I-bands. So we feel our studies have important implications for ALS, which involves altered energy metabolism. Thus far, a common feature of ALS patients is that they have reduced VAP-B levels and abnormal mitochondria in striated muscle and motor neurons. So our model might help explain why motor neurons in muscle are specifically affected in this disease. So while the study focuses on neuron-to-muscle signaling, the neurons also express MSP receptors, and these are likely to play a role. Clearly, there is still quite a bit to learn. So we encourage you to read more about our study in this developmental cell issue.